Well, hello and welcome to all attending the sixth session of the NPRC's 60th Anniversary Scientific Seminar Series. Today's focus is on research in primate reproductive science, which really was a founding core strength of the NPRC's, as from the 1960s to the present day, it's encompassed broad range of topics, including high impact basic science research in primate reproductive behavior, in sexual differentiation of brain and behavior, neuroendocrine regulation of fertility, reproductive tract development, pregnancy, puberty, assisted reproductive technologies, contraception, and of course, much, much more. These fundamental studies in turn have spawned translational research that's only been possible in non-human primates, given the shared physiology between monkeys and humans. A 28-day menstrual cycle, common pubertal control mechanisms, or placental physiology, and also due to the shared pathophysiological states, such as polycystic ovarian syndrome, which we're going to hear about today, endometriosis or preeclampsia. Moreover, NPRC-based research in non-human primate embryology led directly to the seminal discovery of non-human primate embryonic stem cells, which was a critical, if not crucial, antecedent to the derivation of human embryonic stem cells and the maturation of the whole field of regenerative medicine. So these discoveries have also, in more recent times, facilitated the use of cutting-edge transgenic and genomic editing methodologies to generate new non-human primate models of human disease. So the field has come a long way over these many decades and remains a vibrant, exciting frontier of research, highly relevant to human reproductive health. So today we have presentations by three outstanding speakers who will in turn address important issues in basic and reproductive research. Before we start, want to mention one thing is that Dr. Christina Adams Waldorf will not be available for the Q&A at the end of the three presentations, but we encourage you to submit your questions for her that she might be able to answer at a later date. I'd like to encourage you to submit questions for our presenters so that we can have a thoughtful discussion after our presentations, and that questions can be submitted at any time through the chat Q&A function, and will be answered by our presenters in a panel discussion towards the end of at the end of the three presentations. So with that, I'd like to introduce our first speaker. Dr. David Abbott will give our first presentation, Non-Human Primate Models of PCOS Pathogenesis. Dr. Abbott is a professor in the Department of Obstetrics and Gynecology at the University of Wisconsin-Madison. He's also a senior scientist at the Wisconsin National Primate Research Center, where he's been a PI since 1991. Dave has been a pioneer in the study of PCOS pathogenesis, establishing the rhesus macaque as a singularly critical model for this common disorder, and really was the first to demonstrate that prenatal androgen exposure can be a key pathological determinant of the disease. So with that, I turn it over to David Abbott. Dave, take it away.
Many thanks for the kind introduction, John. Hello, everyone. I'm David Abbott from the Wisconsin National Primate Research Center and the Department of Obstetrics and Gynecology at the University of Wisconsin in Madison. I'm delighted to have been invited to contribute to the 60th anniversary celebrations of the NIH-funded National Primate Research Center Consortium. Today, I'm going to be talking about how non-human primate models have been critical for initiating a paradigm shift in clinical thinking about the causes of polycystic ovary syndrome, or PCOS, a subfertility and metabolic dysfunction disorder that affects about 15% of women and contributes to billions of dollars of healthcare costs in the US alone every year, let alone the personal impact it has on women themselves. In these research endeavors, I've been very fortunate to have longtime collaborators and friends, especially Dan Jamezik in the Department of Obstetrics and Gynecology at UCLA, John Levine, Director of the Wisconsin National Primate Research Center, and Jeff Rogers in Molecular and Human Genetics at Baylor College of Medicine in Houston, and all our colleagues who form an integrated multiple disciplinary research team that is key for complex disease research. And I'm also eternally grateful to continued funding from NIH, including P50 Center grants led by Andrea Deneff and John Marshall. Polycystic ovary syndrome. What is it? Well, to be clinically diagnosed with PCOS, a woman must have at least 2 out of the following 3, or all 3. She has to have 2 high levels of circulating testosterone. That means her ovaries are producing too much testosterone. And the accompanying pathophysiology with that is that there's increased LH, or luteinizing hormone, secretion from the pituitary. The second criterion are intermittent or absent menstrual cycles. In other words, there are too few ovulations per year. And the final criterion, polycystic ovaries. In other words, the ovaries growing too many follicles. And you can see that here represented on ultrasound images of a human ovary in the bottom right-hand corner, with the normal ovary focusing on growing one large dominant follicle to ovulation, and the larger polycystic ovary that is growing far too many follicles and not focusing on growing one of them to ovulation. And the accompanying pathophysiology to that ovarian morphology is increased ovarian release of anti-Müllerian hormone, or AMH, that is produced by the individual follicles. PCOS also is a diagnosis of exclusion, but we're not going to go into that today. We're focusing on reproduction. And also, as you heard in my introduction, accompanying metabolic dysfunction, but we're not going to go into that today. We're going to focus on the reproductive aspects. Problem: PCOS origins are really not well understood at all. We do know that it occurs spontaneously in women and in female macaques. In women, it's highly familiar. We're looking into that with an R21 grant in female macaques at present, and that the high testosterone levels are the most heritable trait in women, and we're examining that in our monkeys as well. There are about 22 risk gene candidates for PCOS in women, but they account for less than about 10% of PCOS. And many of them are related to LH action or release, or FSH or ACTH, and insulin receptors. One aspect that the monkey models showed up was that, and now is being borne out by human evidence, is that excess testosterone starting in gestation and fetal life can be key to reprogramming the epigenome in these females, so that regardless of genotype, their phenotype resembles PCOS in adulthood. So PCOS pathogenesis, thanks to monkey models, may have epigenetic, and now may be in mimicking the human condition genetic components as well. Why use female rhesus macaques? Well, as I hardly need to tell quite a few of you in this audience, they share just so many attributes to ourselves, including right down to the actual DNA sequences for genes encoding proteins. So they are most likely to be the animal model that will emulate PCOS in women rather than any of the others. Now, we've been following 3 interlinked hypotheses over the years to examine potential causes of PCOS. The first one, initiated now first published by us 25 years ago, involved giving testosterone, inducing fetal male levels of testosterone in otherwise normal fetal female rhesus monkeys, and producing, when they were adults, the PCOS-like condition, because you can't actually, PCOS is not diagnosed in anywhere but women, so they're PCOS-like. That was followed up 20 years later by a first publication here again at the Wisconsin National Primate Center, showing potentially naturally occurring female PCOS-like condition in our rhesus macaques that may have a genetic basis. And today I'm going to focus on another hypothesis, this time getting down more to molecular action, and first raised by John Marshall at the University of Virginia, that diminished estrogen action in the hypothalamus may be a key pathogenic component contributing to PCOS. So in this talk, I'm going to focus on hypothesis number 2, and that's diminished estrogen receptor alpha expression or activity in the adult hypothalamus may be a key contributor to PCOS. Our hypothesis suggests that leads to insensitivity of estradiol and therefore progesterone negative feedback on release of a key releasing hormone. So the pituitary product is also increased because of that extra stimulation, and that LH stimulates ovarian theca cells to produce too much testosterone. And all this is linked to diminished fertility. I'm going to leave out the metabolic dysfunction for today. Okay, why focus on the estrogen receptor alpha and the medial basal hypothalamus? Well, thanks to elegant studies by John Levine and others in adult female rodents, estrogen receptor alpha, ER alpha, or the gene encoding for it, ESR1, mediates the majority of estradiol negative feedback action from the ovary on hypothalamic GnRH release. And that GnRH release is key for driving the pituitary and therefore ovarian function. Now, this estradiol exerts its negative feedback action on GnRH by binding to ER alpha on neurons, that's nerve cells, in the arcuate nucleus of the medial basal hypothalamus, as illustrated up here. If we're looking at this diagrammatic medial basal hypothalamus, and here we have ER alpha receptors on neurons involving kisspeptin, neurokinin B especially, but also NPY, dynorphin, GABA, as shown by A. Terasawa and others here at the Wisconsin Primate Center, all housed within the arcuate nucleus here, and all synapsing onto GnRH neurons to control the release of gonadotrophin releasing hormone that's key to be released in a pulsatile episodic fashion to drive luteinizing hormone and FH release from pituitary gonadotroph cells that also have ER alpha on them, to drive hormone release and mature egg development in the ovary. And this homeostatic condition is controlled by estradiol negatively feeding back and binding to ER alpha on those neurons that control GnRH in the arcuate nucleus, but also in primates, certainly onto ER alpha on the gonadotroph cells as an extra layer of negative feedback. Okay, now the problem is in primates, including ourselves, the mediating estrogen receptor for estradiol is unknown. So our hypothesis was to looking at 2 aspects here, that knockdown with a viral vector of estrogen receptor alpha in the adult female hypothalamic arcuate nucleus alone will partially diminish estradiol negative feedback restraint on GnRH release in adult female macaques, as illustrated here in a diagrammatic version of that hypothesis, that if we infuse a virus that carries silencing RNA for the gene encoding ER alpha, so that our messenger RNA is never translated in or is hugely diminished in its translation to the protein ER alpha in the arcuate nucleus, we will then diminish negative feedback regulation of estradiol on GnRH. So GnRH levels will go up. That will lead to increased LH release from the pituitary, as I illustrated earlier, increased ovarian androgens because of LH receptors on theca cells, and menstrual cycle dysfunction. These are all aspects of PCOS reproductive phenotype that I mentioned at the beginning. Does this happen? And the experimental design, just to let you know here, is we used adult female rhesus macaques, about 11 years old. That's in the prime reproductive years of their lives, about 8 kilos in body weight. That's a nice average body weight, ovary intact, exhibiting menstrual cycles before we started. And we had two groups, one that received a scramble virus where the silencing RNA was targeted against no protein coding gene. And the other group, and there were 5 controls where we had 6 adult females, where now the viral vector has silencing RNA targeted specifically against the gene encoding for ER alpha, and therefore preventing translation of the protein to its viable receptor. We pick up the story in this 3-year study, 36 months. This is not a rodent study where it's all over and done with in weeks. In primates and in women, this is a long-term issue. We pick up the story looking at menstrual cycle duration and hormone assessments 18 months into this 36-month study. And we also gave an ovarian androgen stimulation test in the last few months of the study here, and then looked at tissues at necropsy at the very end. This is just to show you at surgery, when we infused the virus, we knew during surgery that we'd accurately targeted where we wanted it to go. Now, this is a coronal section of an MRI scan in mid-surgery, in this case, of a rhesus monkey actually getting the silencing RNA for ER alpha knockdown. And you'll see the cannula tracks here in this coronal image, the brain here in coronal section through the hypothalamus area, and the whole rhesus head here. And you'll see those cannula tracks. The cannula is not present at this time when the animal is in the MRI scanner, have infused the viral vector, also with a contrast agent for MRI, into the ventral medial nucleus, not our target for today, but also the arcuate nucleus on both sides of the hypothalamus. So we did target where we wanted to go, and we knew that during surgery. And here's the anterior pituitary sitting below. Now, this is the posterior pituitary that shows up dense white, as it does in all images. It's not spillover of infusion down there. That's just its natural presentation. Okay, 3 years later, at the end of the study, did we knock down ER alpha expression in the arcuate nucleus as we proposed? Well, in this example, in the panel on the left here, you see one control female, and you'll see stain from a monoclonal antibody raised against green fluorescent protein. That we did indeed infect cells because those viruses also contained a promoter for green fluorescent protein. So any cells we infected will stain up brown here, and there they are. This is in the caudal region of the arcuate nucleus down here. And if we look for using a monoclonal that stains against ER alpha, and you'll see here these speckledy brown dots show ER alpha expression in the control. And in this enlarged image here, you can see the nice nuclear staining of ER alpha, as you would expect. What about an example from a knockdown monkey? Well, if we look in the same caudal region of the arcuate nucleus, no obvious staining. When you see up here in the enlarged image, there's very, very light staining there. We've achieved a significant loss of ER alpha protein expression in this area. This is just one section here, whoops. But if we look at the panel on the right, where we quantified, and we'll just look at the arcuate nucleus here, and in controls in blue and our knockdown animals in orange, you'll see we got significant knockdown of ER alpha through the arcuate nucleus in our knockdown monkeys. Okay, what did that do to their menstrual cycle parameters? Again, the controls are in blue, our knockdown animals are in orange, and you'll see menstrual cycle duration is not changed. If you look at follicular phase duration, that's from menstruation up to ovulation, no different. And you look at luteal phase days duration, no different. And this is between 18 months and 36 months into the study. And 2 out of 5 of the controls became anovulatory and 1 of the 6 knockdowns. So these are the remaining animals here. So duration of the cycle didn't change, but one part of the luteal phase did in terms of function. And we picked that up by looking at luteal progesterone levels here. We can see, and again, our knockdowns in orange, whether we're looking at peak luteal progesterone, mean luteal levels, or area under the curve luteal progesterone, our knockdown animals show significant diminished luteal phase progesterone, which we know certainly in women can lead to inadequate support of early pregnancy. And one of the common causes for this is suboptimal gonadotrophin support of that dominant follicle that becomes the corpus luteum during the follicular phase. So were there disturbances in function during the follicular phase, even though it too was of normal duration? The short answer is yes. If we look at the top right-hand panel now in this follicular phase, you'll see LH levels are elevated, just as we hypothesized. Did androgen levels elevate at the same time in the follicular phase? Testosterone, not significantly, but it's an immediate precursor that is metabolized to make testosterone. Androstenedione significantly was in this last 18-month period of the study. If we look at FSH, the other gonadotrophin, not different, and that's not too uncommon in PCOS as well. Estradiol, also not different, and that's very common in PCOS too. But unlike PCOS, where AMH and those orange bars should be elevated, it's not in our animals, suggesting it's unlikely that they have polyfollicular or polycystic ovaries. Now, that androgen, that elevated androgen, is it ovarian? And we submitted these animals during the follicular phase or anovulation during the same time period towards the end of the study, about 2.5 years into it, to an ovarian androgen stimulation test. We inject chorionic gonadotrophin intramuscularly, and 24 hours later look at testosterone and androstenedione, 2 major androgens that will be stimulated by ovarian theca cells by CG binding to the LH receptor. And you'll see in both cases, significant for testosterone, trend for androstenedione, elevations over controls. This is ovarian androgen excess, just as in PCOS. But we don't have polycystic ovaries. As you'll see here, our knockdown females are in the middle, and you'll see from the histology at the bottom too, their proportion of growing follicles that contain an oocyte each, primary, preantral, and antral, mimic that in our controls. And both are typical of age-similar non-study females here on the far right. These ovaries aren't any larger, and they haven't committed to more growing follicles. We don't have polycystic ovaries. One interesting thing that did turn up that we weren't expecting was endometriosis. It's a rare finding with PCOS, but 5 out of our 6 of our knockdown animals did have endometriosis, and one of them had an endometriosis-associated ovarian cancer. And Manish Patankar in obstetrics and gynecology, Shannon Rush in gynecological oncology, and Heather Simmons, our head pathologist at the Primate Center, all following up on this aspect. And just to say, yeah, this also induced a metabolic defect, but that's for another time. So in summary, pulling all this together, if we look at our knockdown animals here in red, how did they compare to our gold standard gestational T exposure PCOS model? Well, they did induce ovarian androgen excess. Yes, we didn't get intermittent or absent cycles, but we did get luteal insufficiency that would compromise fertility, and there was no polycystic ovaries. We did get androgen excess. We did get LH excess. but we don't know much else about that reproductive phenotype as yet. So in summary, in ovary intact adult female rhesus monkeys, partial estrogen receptor alpha knockdown in the hypothalamic arcuate nucleus results in mild PCOS-like reproductive phenotype. And this is consistent with the John Marshall hypothesis that we started with in terms of luteal insufficiency, LH hypersecretion, ovarian testosterone excess. So in conclusion, as we hypothesized, we identified ER alpha in the arcuate nucleus of the hypothalamus as an important molecular mediator of estradiol negative feedback regulation of gonadotrophin release in a female non-human primate, and likely in women as well. So we may have got our estrogen receptor in primates now. Diminished ER alpha expression in the arcuate may contribute to a PCOS-like phenotype, and a degree of negative feedback function remains, however, likely mediated by ER alpha escaping knockdown in the arcuate, as I showed you, and on anterior pituitary gonadotropes. And the potential clinical translation here is that brain, not somatic delivery of estradiol, may alleviate PCOS. And this was first developed and shown using a synthetic compound that's converted only to estradiol in the brain in female rats by Dr. Merkenthaler, and is now being continued by him and Dr. Lacruz in ongoing trials in marmoset monkeys. Exciting possibilities there. Non-human primate models leading the way again. So thank you for listening, and I'll be only too happy to answer questions in the Q&A session. Thank you.
Thank you, Dr. Abbott, for a wonderful talk. Given our delay in getting started, we're going to jump right into our second presentation, non-human primate models of reproductive health and development.
Thank you, John. And I'd especially like to thank the organizers for putting together an amazing 60th anniversary of the Primate Center's seminar series that has highlighted not only the amazing work that's been occurring at the National Primate Centers, but also all of the work happening globally with non-human primate models for human health and disease, and highlighting all of the work remaining to be done and the true importance of non-human primate models for that work. So today I'd like to highlight some of the work that's been occurring and the work that remains to be done in maternal and infant health issues. And while we've made tremendous strides in human health overall, and we've heard a lot about those over the last few weeks, I argue that there's so many issues still remaining, especially in female reproductive health and maternal health and infant health, that's resulting in mortality, resulting in infant loss that we don't have solutions for yet. And we have a lot of effort to put in to get these answers. When we look at the CDC's website for maternal and infant health, they highlight these issues where clinicians are working and where research remains to be done, and they range from pregnancy complications to maternal mortality and preterm birth and SIDS. And so we think in the modern world that we've solved many of these problems, and certainly we're better off now than we were 100 years ago, but the numbers are still striking for the issues that we have in maternal and infant health. And so when we look at maternal mortality globally, what you see is, again, just striking numbers of mothers that are lost due to complications during pregnancy and complications during delivery that range from hemorrhaging to ectopic pregnancy. And so while the US is certainly better off than many other nations, we haven't solved this problem completely. And there are still concerns and clinical treatments that are needed. When we look at infant death, these numbers are absolutely unbelievable, that we are still losing almost 3 million infants every year to stillbirth and almost 3 million infants to early neonatal deaths. And these deaths occur often within the first week. And many of these deaths are associated with a lack of prenatal care and a lack of clinical care. But this window of pregnancy isn't very long. And so it gives us a very short time frame where we need to focus on interventions and understanding health and disease and preventing these losses. And again, while I give you these global numbers, there's still risks in the US, and there's still states that are at much higher risk for infant loss neonatally than other states. So a lot of work left to be done. And one of the things I want to highlight is, we think we've solved things, and then we have a media burst a week ago about SIDS, which many people don't think about. We think about SIDS as a problem that our grandmothers faced, but SIDS is a very real problem, and we have mothers losing infants due to SIDS. And this media storm released that SIDS had been solved and there was a breakthrough and there'll be a cure. And the scientists are rapidly responding with, wow, this is a great discovery. Even in the paper itself, it marked that it was a first step and a first step to a biomarker and a possible intervention. So we have a lot of work to do for this type of disease. Non-human primates have contributed tremendously to maternal health and infant health. And a few of the species are highlighted here. And I just picked a few to highlight. And in a recent review article in the American Journal of Primatology, they do a nice job of highlighting all of the species that have been involved and the types of topics ranging from birth control, efficacy and toxicity, to endometriosis, to preeclampsia, which we heard a little bit about earlier, to infectious disease outbreaks that are current and recent. And so things like Zika disease, Zika infection, which had horrific impacts on fetal health, and looking at using non-human primate models to look at vaccine treatments and preventions for Zika. And now there's still ongoing tests in non-human primate models to look for solutions to hemorrhaging during delivery so that women aren't lost due to hemorrhage. I just want to highlight a few contributions that species have made, and I'm going to not highlight rhesus macaques today because we've talked about them a lot in the seminar series, and I want to highlight a couple of the other species models that are highlights of Southwest Primate Center, even though not all the work was done here. But the first is the baboon and the contributions that it's made to premature infant. And so the first example I'll show is the baby Faye heart transplant, in which baby Faye was a premature infant and she received a baboon heart. And while she didn't survive, she survived much longer than previous attempts. And she was the first in a series of steps to get closer to the ability to do heart transplants for premature infants that were at high risk for death. And so baboons contributed tremendously to that ability to do those interventional surgeries. One of my favorite examples from baboon premature infant modeling work is the development of surfactant as a treatment for infants with premature underdeveloped lungs and ventilation to safely maintain oxygen levels for those infants. And this is work that was done at Southwest Primate Center. And again, we think of this as having been done a long time ago, but this work was done in the late 90s and early 2000s and tested, again, efficacy and toxicity and ability of surfactant to protect and develop infant lungs. And one of the reasons this is my favorite is because it personally affects me and saved my daughter, who was born in 2003. And she received both ventilator treatment and surfactant. And again, in 2003, it never even occurred to me that this wasn't a standard clinical practice that existed for a long time. It was fairly new, and now is standard practice in saving millions of infants every year. One of the topics that's particularly important in non-human primate research and in maternal and infant health is the idea that we can look at developmental origins of health and disease. And this is also known as the Barker hypothesis. And this hypothesis states that things that happen in early development, things like infection and low birth weight, impact later health. And the later health can range from aging, which is a topic I study now but won't talk about today, to other disease risks like the female's ability to reproduce as an adult, polycystic ovarian syndrome, stroke, hypertension, obesity. And I'll talk about obesity in a little while. But we can focus on non-human primate models to study this relationship between fetal development and adult health because they mature more rapidly and because we can manipulate and control that early environment and the risks that are posed to those infants during development. And so again, the baboon has played a critical role in studying developmental origins of health and disease. And the first study I'll highlight actually was a field study at the Amboseli Project. And this is led by Jane Altman and Susan Alberts. And in this study, they found that early adversity to the mother, so a female that has as an infant a loss of her mother or a loss of a sibling, or even worse, the loss of both, are much more likely to lose their infants when they are adults. And so this adversity that is faced in the mother while she's an infant significantly impacts and programs her ability to reproduce as an adult. And this is a significant health outcome. And of course, there's been decades of research at Southwest with the baboons led by Dr. Nathaniels and his team, looking at the impact of a number of factors during prenatal development on adult health, including things like if the mother is nutritionally restricted and the impact on those infants on their glucose metabolism as infants and juveniles and then as adults. And they have found significant impacts on these health challenges to the adults and the reproductive health and infant health. And so they're starting to tease out the mechanisms that contribute to those developmental origins of disease. I'd like to switch over to the marmoset, which is the model that I work in and have worked in for the last 20 years. And marmosets may not be as familiar to everyone, so I'll just introduce them a little bit as a primate. So they're small, squirrel-sized primates. So they are native to Brazil, and the most common common marmoset used is the common marmoset, Callitrix jocus. And they, one of the reasons that they're advantageous for a number of biomedical questions, but certainly for reproductive health and infant health, is that they have a relatively rapid growth and a short lifespan. So they wean as early as 3 months of age. They reach full sexual maturity and adult size by the time they're 2. Average lifespan in most captive colonies is around 6, which is longer than it is in the wild. And the oldest marmoset reported was 22 years. And so I already outlined developmental health and developmental origins of disease. And so having this short-lived, rapidly producing primate is advantageous. And by rapidly reproducing, they have a short gestation of about 5 months, so about 145 days. They are litter-bearing primates, so they were considered obligate fraternal twinners because the predominant litter size is twins, but they can have litters that range in size from 1 to 5. And they do not have post-delivery amenorrhea, so they cycle again 2 weeks after delivery and can become pregnant again almost immediately, and so they can produce 2 litters a year. And one of the things that's fascinating and perhaps really important when we think about early life adversity and the impact on later adulthood health is the fact that marmosets are cooperative infant caregivers. And so juveniles care for the infants, and then males actually become the primary caregiver after about the 2nd week of life. And so the moms are still nursing, but the males become the primary carrier and security and safety for the infants. So when we work with marmosets, one of the advantages is we can do a number of the things. They're small, so they're easy to handle and handle without anesthesia. And so what you see is a little bit of an older image, but this is Dr. Tardif and Donnaline Cologne doing an awake ultrasound. And you can see the marmoset is happily eating fluff off of a stick. And she is pregnant with twins. You can see 2 little heads here. So we can ultrasound them all through pregnancy and follow infant development and prenatal growth. And then once they're born, the infants can be removed for measurements and behavioral assessments and returned to mother without rejection. And one of the other things we can do is if we're interested in questions such as obesity, the marmosets are small enough to fit into a quantitative magnetic resonance machine, a QMR, to look at fat and lean mass. And so this is, again, an awake test. The animals crawl into the tube. It's a 2-minute MRI, and we have fat and lean mass measurements. So again, an advantage of working with the marmoset model. Today, I'd like to highlight a few of the projects that we've been doing with marmosets as models to examine questions in developmental origins of health and disease, both that impact maternal health and reproduction, as well as early infant health and potentially later health. And the first that I'll start with is the Womb to Womb project that was led by Dr. Julianne Rutherford at the University of Illinois, Chicago, and completed at the Southwest National Primate Center with the marmosets here. And we were interested in looking at marmosets to examine how that prenatal environment impacts a female's reproductive ability when she reaches adulthood. And one of the reasons we began exploring this topic is that Julianne found when we look at females that are born as triplets rather than as twins, they're 3 times more likely to lose their own offspring to miscarriage and stillbirth. And so while females that are born as twins do lose offspring as adults, they lose offspring significantly less than if they're born as triplets. And so we are interested in whether this is a factor of being a triplet, if it's a factor of birth weight, and how that impacts uterine growth, ovarian cycling, and maturation of their own infants. And so we studied mothers across multiple generations. We followed females for multiple generations, and we're still working through all of the data and what it means for the marmoset and for marmoset infant health. But one of the things that we did find that's really important is that while being a triplet significantly impacted risk of infant loss during pregnancy, separately, independently, maternal birth weight is linked to increased risk of loss. And so it's not the weight of a mother while she's pregnant or even immediately pre-pregnancy. It's not the amount of weight that she gains during pregnancy. It's the weight that she was born at, of which she had no control as an individual, right? So that's what impacts infant loss. And so one of the things that's important when we think about this translationally and clinically is that we need to reassess the narrative that's being promoted to women in the clinic, that pregnancy outcomes are entirely associated with their health and their choices during that pregnancy. And while those factors are important, and they certainly pose a risk to the pregnancy, they are not the only thing that impacts the pregnancy. And this is particularly important as we enter this political climate in the US, and there will be ramifications that impact women and women health outcomes. The other topic that we worked on and spent a great deal of time working on is the idea of maternal obesity and how maternal obesity is linked to maternal health and infant health. We all know that in the US, we're faced with an epidemic of a health crisis of obesity, and so much so that the CDC changed their color coding between 2007 and 2020, because there are no longer states that are reporting less than 20% obesity in their populations. And in epidemiological studies, we find that maternal obesity in humans is linked to an increased risk of infertility, an increased risk of pregnancy loss, and then increased risk for infant development of obesity. But again, the mechanisms are unclear, and the risks to the mother and infant really remain unclear. And so we worked on the marmoset model of obesity, and they are a particularly good model for this because they become spontaneously obese. So this is a twin set where the male is obese and the female is lean. So we have spontaneous obesity, and we have the ability to induce obesity through high-fat feeding. And so we looked at mothers that were on a high-fat diet and exposed and were obese, and we see no difference in ultrasound of prenatal growth. And so this is crown rump length growth, and this is abdominal circumference, and there's no significant differences during prenatal development. But we do see differences postnatally. So we see a difference in trajectory of growth in infants that's related to birth weight and then weaning. And so infants that go on to be obese as adults split in trajectory at about 3 months of age. And these infants were more likely to have been of the higher birth weights, and that higher birth weight is linked to maternal obesity. And what we find if we focus in on feeding behavior in early infant care is these infants are weaning sooner. So they're exposed to first day of solids sooner. And those infants are also off of the caregivers, both the older siblings and the father, much sooner. In mother care, maternal care doesn't differ, but it's the carriers. And so one might wonder how this is translationally relevant, and it's translationally relevant because we clinically often suggest to mothers that they start weaning sooner, they start introducing solid foods sooner because it's easier and it gets them to sleep through the night. And again, currently, this is potentially a really large problem as we're facing supply chain issues with formula. Clinicians are recommending, in some cases, that parents begin introducing solid foods sooner, which could directly impact obesity of these infants. So as a final note, I just want to highlight some things that we need to keep in mind when we study animals like marmosets, and this is their natural biology and a potential conundrum. So marmosets are chimeras, and I haven't mentioned this yet, and we often don't discuss it because it's a potential problem for them as models. They give birth to fraternal twins, and these twins share a placenta, a bidiscoid placenta that's connected through anastomoses. And during development, stem cells exchange across these anastomoses. And so we have chimerism, we have natural chimerism that occurs in most mammals, known as microchimerism, where there's an exchange between a mother and the fetus. And then in marmosets, we have this twin-to-twin transfusion during development. And it's unclear how this twin-to-twin transfusion then impacts microchimerism and health of the mother. And it's unclear how this impacts health of the infants that are developing. And when we look at prevalence of chimerism across tissues, this data is a little controversial. It's older, it's microsat data, but we see chimerism throughout the tissues. And we don't know what this means for marmosets as reproductive models. But I actually argue that this makes them a very strong model for very specific questions. We are seeing an increase in multiple births, especially in women over the age of 40, due to IVF. And we have absolutely no idea how, if there's an impact of chimerism, and we know humans can be chimeric, we don't know if there's changes in prevalence of chimerism, how that's impacting infant health, and how that's impacting adult health when these infants mature. And so we may be exacerbating some of the problems with human reproductive health through IVF and potential impacts like chimerism. And so this is one of those things where knowing your model can help the modeling in the questions. And so we're hoping that marmosets can continue to contribute to these types of questions. But overall, the underlying message is that rhesus macaques may be better for certain questions and marmosets are better for others. And we need to have all of these models available to answer the questions for human health. So I'd like to thank my team, both past and present, and the funding for the research that was highlighted today. And thank you all.
Thank you very much, Corey. That was terrific. I'm going to try to move on quickly. Our next speaker is Dr. Christina Adams Waldorf, who is a professor in the Department of Obstetrics and Gynecology and adjunct professor in global health at the University of Washington School of Medicine. Dr. Adams Waldorf is going to be talking to us today about influenza disease pathogenesis in a pregnant pigtail macaque model.
Hello, my name is Christina Adams Waldorf. I'm a core scientist at the 1PRC. I'm an obstetrician by training, and I study reproductive infectious diseases and therapeutics in the non-human primates, specifically the pigtail macaque. So today I would like to share with you some of the work that we are doing in our program, and specifically how we are studying the pathogenesis of influenza disease in a pregnant pregnant pigtail macaque. This diagram kind of gives you a sense of all the different areas that our lab is doing work in. It began with studying preterm birth, bacterial virulence factors, and the pathogenesis of infection-associated preterm birth, as well as therapeutics in pregnancy to block preterm birth. With Zika, we began studying Zika virus, perinatal viral infections, and now most recently with influenza A viral infections in pregnancy. We also study fetal injury, including lung, cardiac, brain, and placental injury. So today we'll talk about influenza virus. So influenza A virus is known for having enhanced disease in pregnancy, and in several pandemics that has been clear, especially in 2009 with the H1N1 pandemic. Pregnant women were approximately 1% of the US population, but accounted for nearly 6% of deaths in the US. In addition to maternal mortality, preterm birth, stillbirth, and neonatal death can occur from infection. We need a pregnant non-human primate animal model to test universal influenza vaccines, because testing in a unique vulnerable group is a benchmark of vaccine success. I'm going to talk a lot about type 3 immunity today, so wanted to make sure we were all on the same page. Type 3 immunity is characterized by IL-17 and IL-22 expression, the transcription factors ROR gamma T and ROR alpha. It recruits neutrophils and stimulates epithelial antimicrobial defenses at infection sites. And some key type 3 immune cells I'll talk about today are the Th17, the Tc17, and the gamma delta T cells. Well, normal hormones in pregnancy tend to cause a lot of immune dysfunction, including reductions in Th17 cells and an increase in T regulatory cells. So first, high progesterone and estrogen in pregnancy will drive down ROR gamma T expression and Th17 cytokine secretion and proliferation. The high human chorionic gonadotropin, estrogen, and progesterone hormones will enhance FOXP3 expression, expansion of Tregs, and immunosuppressive function. And both of these together will help to dysregulate the Treg Th17 balance in pregnancy, which has been associated with enhanced influenza lung disease in mice. Our hypothesis for this model was that an aberrant type 3 immune response during an acute influenza A virus infection in pregnancy would lead to a broadly dysfunctional innate and adaptive immune response that prevents viral clearance and enhances risk for maternal death and stillbirth. The model was an acute influenza A virus infection that spanned only 5 days, with inoculation on day 0. Today, I'll only show data from our H1N1 experiments. Day 3 is particularly important because this is very Th17 relevant time point. Day 5 is what we think of as peak pathology from other experiments in the Fuller laboratory. And that's when we performed cesarean section, dam, and fetal necropsy. Bronchoalveolar lavage was performed at baseline day 0, day 3, and day 5, and blood and nasal swabs kind of throughout the study. So the first question is whether pigtail macaques become ill from influenza A virus disease, and they do. The pregnant animals, the first 2 that we did, both had fever. One developed significant pneumonia and cough with a significant weight loss and increase in respiratory effort. In the first 3 non-pregnant animals that we did, 2 were very mildly ill, and the second one had, the second one had a weight loss of almost 9% by day 5. So yes, they do become ill. And when we looked for influenza A virus viral RNA using qRT-PCR, we found viral RNA fairly ubiquitously in nearly all of the tissues that we tested, both in pregnant females and non-pregnant females. But what was interesting to us was that we found viral RNA in the placenta and the fetus, because maternal fetal transmission is not thought to occur with influenza A virus. At least that is what has been reported in the human literature. But of course, no one has looked as extensively as one can in a non-human primate model. Interestingly, we found extrapulmonary disease only in the pregnant animals. In the first 2 pregnant non-human primates, we found evidence for T-cell infiltration of the lungs, the heart, and the spinal cord, viral myocarditis, both brain and spinal cord injury. During the early phase of this model, we asked for help from the pulmonary and critical care medicine team at the University of Washington and asked them what was the best way to assess pulmonary physiology. And they said that they would use a human pulmonary function testing protocol that involves an inspiratory pause. And what this is able to do is to assess oxygenation with a PF ratio, which I'll get to in a moment, which is the partial pressure of oxygen divided by the fraction of inspired oxygen, also an alveolar arterial gradient, several measures of ventilation, and measures of pulmonary mechanics. And in this second column here is what we would expect in the face of lung injury. And I don't have time to tell you all about this pulmonary function testing. We are planning to write this up as a protocol paper that can be used by other primate centers if interested to study pulmonary physiology in mechanically ventilated non-human primates. And this would be very useful, I think, with viral respiratory infection models. So here, if we're looking at the PaO2 or FiO2 ratio, the PF ratio, normally the values are greater than 400 in humans. And we see that that's true in one of the pregnant animals and one of the non-pregnant animals. But in several of the pregnant animals, they had slightly lower to start with PF ratios. But then with influenza A virus infection on day 5, we got a drop in the PF ratio for all animals. But in 2 cases, the drop was particularly significant. And it actually crossed over a threshold for acute respiratory failure. And so this is kind of demonstration of a novel application of human pulmonary function testing protocol in mechanically ventilated macaques that we hope will give us a more quantitative outcome or measure of pulmonary mechanics, oxygenation, and ventilation that we can then correlate with our immune correlates of disease and also potentially anchor our single cell and bulk RNA-seq studies. Now I'd like to show you a little bit of our flow cytometry data. And this is from day 5 in the lung and in the paratracheal lymph node. And although the levels of CD4 and CD8 T cells are often fairly similar, but sometimes higher, there were certainly greater activated CD4 and CD8 T cells that are HLA-DR expressing in the lungs and paratracheal lymph nodes of the pregnant versus the non-pregnant animals. And that's what's shown here in these kind of purple columns. Pregnant here is in red and blue is non-pregnant. Both groups are infected. And now showing the Th17 response in the bronchoalveolar lavage, this appears to be impaired on day 3, right here, with a subsequent surge on day 5 at a time when the Th17 response has returned to baseline in non-pregnant animals. So the temporal time course here, the Th17 response looks abnormal. We don't yet have enough data to evaluate the TC17 response. Certainly it appears that this might be somewhat impaired on day 3, but we don't know what's happening yet on day 5 as we don't have enough data. We do have more data on T regulatory cells, and so wanted to give you a sense of what's going on here. And here we find in our Treg cells, we find higher frequencies in the bronchoalveolar lavage on day 3, and inappropriately higher levels of Tregs that express TGF-beta1 in the blood and bowel of pregnant non-human primates on day 3 and day 5. And this is at a time when pregnant non-human primates are clinically much more ill than their non-pregnant counterparts. So in summary, we have evidence of differences in disease severity and immunological landscape for pregnant non-human primates that are infected with influenza A virus 2009, the H1N1 pandemic strain. There's preliminary evidence of aberration in the temporal course of the Th17 response in pregnant versus non-pregnant animals. Infection with the IAV H1N1 virus results in decreased measures of oxygenation in the pregnant non-human primates versus the non-pregnant animals. Pulmonary function testing was successful in ventilated non-human primates, and we believe this is the first time this has been described, and we'll write this up. Maternal fetal transmission of influenza A virus is a phenomenon that was not thought to occur, and at least by qRT-PCR, we have some evidence for this, and we'll follow this up with immunohistochemistry and in situ hybridization. Next steps in our model are to expand our gating from past experiments to more fully analyze the type 3 immune response from our flow and ICS data. We have correlations of pulmonary function testing data that we would like to do with our flow cytometry, ICS, cytokines, and qRT-PCR data. We'd like to determine the immune correlates of disease, and we have single-cell RNA-seq and bulk RNA-seq analyses that are in progress, thanks to a collaboration with Dr. Michael Gale and the Seattle Genomics Lab at the WAN PRC. This is the wonderful team that made this happen. Orlando Cervantes is the graduate student that is spearheading this model. I would like to thank everyone on the team, specifically Hazel Huang, Matt Dakanai, Yenyan Wu, Nicole Bird, Vivian Lee, and Jeff Munson for the amazing data management and data analysis. This would not have been possible without a collaboration with Dr. Deborah Fuller, Thomas Lewis, Megan Fredericks, and Sandra Dross in her laboratory, and the virology and immunology core at the WAN PRC were absolutely critical for doing all the flow cytometry and ICS work. Dr. Melissa Berg, a veterinarian at WAN PRC, and Brittany Curtis were the ones who were able to implement the pulmonary function testing protocol that's done in humans and adapt it to non-human primates with the help of Dr. Sid Kapnadak and Dr. Bill Altemeyer from Pulmonary Critical Care and Sleep Medicine. Connie Fountain and Solomon Wangari were the research scientists at the WAN PRC that implemented all of these studies. Chris English and Audrey Baldessari and Raj Kapoor were instrumental for the necropsies and histopathology analysis. I'd like to thank our funding, as well as the UW IACUC and Environmental Health and Safety. Thanks again to the organizers of the 60th NPRC Symposium Series. Thank you to all the speakers and the attendees, and to John Levine for moderating today, and Leslie Tate for organizing everything and kind of keeping us on track. Thank you, everyone, and hope to see everyone soon in person.
Okay, thank you so much, Dr. Adams-Waldorf. We have time for questions, at least for our first two presenters. So I want to encourage all of you to submit your questions ASAP right now as we speak. And while you are doing that, I will go ahead and get the ball rolling with a couple of questions for Dave and for Corey. Let's roll the dice here. First one is for you, Corey. It's really a fascinating finding that maternal obesity is linked with reproductive compromise in offspring. I'm just wondering if you could speculate on what mediates this apparent programming effect of maternal obesity.
That's a great question, and it will be full speculation because we're still working through data for both the maternal obesity project and the womb to womb. And this is where they connect, and where we think that the mechanisms connect based on litter size. So our obese females are more likely to superovulate and have larger litters. And so then those females are being born as triplets, and they may have a higher likelihood of then growing up to be obese themselves because their mothers were obese, and there's differences in feeding mechanisms. But they also are triplets. And so then they have impaired reproduction as adults. And so we're working through some of the mechanisms we know and have published on epigenetic changes in the placenta during development of obese females. And we also have some evidence that I didn't mention today because we're prepping it for publication, but Julian Rutherford and Tony Ziegler have been working on chorionic gonadotropin and the luteal placental shift, and finding that that is significantly different in triplets. And that again is linked to obesity. So I think it's not going to be one answer, but perhaps Dave's mechanisms might be something for us to look at, because the things he was describing could very well play a role in litter size and in obesity for the marmosets, and then translationally for humans.
While you're speculating on that, can you also add whether you've seen the same sort of programming effect on males as well as females?
That's a great question. And everyone always asks us to look at boys, and we're apparently our bias is a little opposite of most researchers, where we focused on the girls, but we do see differences in obesity development. That males will also go on to be obese, but we don't know what their fertility looks like. And if we don't have any data from the health records that suggest those males have a reduced fertility, but it's very possible that we have the epigenetic shift impacting males as well.
Is there human data that address that possibility in either sex?
Yes, but a lot of the human data focuses on hormonal imbalances. And so males that are obese tend to have altered cortisol and altered metabolism, and therefore altered testosterone, which then impacts fertility. And they also have blood pressure issues, and males with blood pressure issues are on blood pressure medication, which then impacts testosterone and fertility. So it's complicated in humans. And same for females. We have had cases in the marmosets now where we have females that are larger than just obese. So our obesity characteristic was 450 grams and above. We have marmosets females that are now 600, 650, 700 grams. So they're twice the size of what we used to consider a normal marmoset. And these females don't become pregnant at all. And if they rarely become pregnant, they miscarry early in pregnancy. And we don't know what those mechanisms are and how that relates to morbid obesity and human reproduction.
Thank you. Here's one coming your way, Dave. So you didn't have time to go into the metabolic features of PCOS and whether you conferred any of those or any of those phenotypes appeared in the estrogen receptor alpha knockdown animals. Do you want to take this time to elaborate on what sort of metabolic features of PCOS that also appeared?
Thanks, John. Yeah, you picked up a very good point there, that the other nucleus we knocked down ER alpha in was the ventral medial nucleus, which plays a big role in regulating energy homeostasis. And in our monkeys, they certainly gained more weight than the controls. They had diminished locomotor activity because they were wearing collars during the morning, the first 5 hours or so in the morning, but not during the rest of the day. So they gain weight more, they are less energetic. And if we look at a postprandial energy measure, basically, long story short, we put them in a metabolism cage and measure the ratio of oxygen to carbon dioxide following a meal, and they exhibit less heat generation during a meal. That's what we all do, but the controls release more heat than the ER alpha knockdown, suggesting they're expending less energy, and that's probably what's leading to their weight gain. but they're not eating anymore. If you look at the calorie intake, it doesn't change to the controls. So when, again, the monkeys are showing that, don't think of people who are putting on weight as they're just eating more calories and they can't control themselves. The monkeys are putting on weight, but it's because of a physiological change. Estradiol is less active in parts of their brain, so they can't control their energy homeostasis. That's just like PCOS, may be of relevance in Corey's models too. She'd have plenty of room to have a look there. I don't know, but it certainly opens the door there, showing that it can be very much a physiological issue that could be addressed by, as I said at the end, giving perhaps brain directly into the brain. Okay, you don't inject it there. You take it as a pill, as Dr. Merkenthaler is working on, and it gets across into the brain and only becomes estradiol in the brain, because that's where the enzyme that breaks up that compound exists, nowhere else. So it's an interesting insight that the monkey model's given us.
I'm wondering if you could also speculate on the set of PCOS-like symptoms that you're observing in the ER alpha knockdown animals versus the larger set of PCOS features that you've identified as naturally hyperandrogenic female macaques with PCOS-like syndrome. How do they compare, and why would one be a milder version of the other?
Well, with the knockdown animals that I went through in the talk in some detail, the knockdown was not complete. So it could be if we got a more complete knockdown, they'd have a stronger phenotype, but the phenotype was the high testosterone. That's a key part of PCOS. There is an indication of the neuroendocrine defect there as well as there is in PCOS, but we didn't really get a polycystic ovary. That didn't turn up. Whereas and AMH levels were not elevated. Whereas the naturally occurring PCOS-like animals, they have the high testosterone, they have the neuroendocrine defect, but they do have elevated AMH, and in one population they have enlarged ovaries, suggesting that they've got more of the complete package. And if we look at the very first model we started to work with, the fetal gestation testosterone, they have the whole 9 yards, except they don't have, again, the polycystic ovary. So it may say the genetic basis for PCOS may have a lot to do with the polycystic ovary, but maybe you can have physiological defects during gestation. And Corey's given rise to some causes that can lead to that. Or you can have an estradiol deficiency in various ways, and you can get to a close mimic of PCOS without the polycystic ovary.
Okay, I'm going to ask both of you a question that's been asked of a number of speakers in each of these sessions, and that is, why non-human primates? Why did you select a non-human primate model to study your experimental questions? And why did you choose those that you're using in particular as opposed to other non-human primates? What makes non-human primates critical as a model for the questions that you're trying to answer?
Okay. The big one for us, because we're looking at genetics, they are over 97% identical in the gene sequences and genes coding for proteins. That's an amazing similarity. But if you look at their developmental history from gestation all the way through to puberty and into menopause, they mimic us. They're menstrual species. They have one ovulation a month. They give birth to one infant. That's why we went for the macaque for PCOS. Corey is right. You have to choose your animal model for your human issue and medical issue you're trying to deal with. And for PCOS, those are some of the reasons, as well as metabolic similarities, that the rhesus macaque certainly made it. But if it was another human disorder we'd be focusing on, I might choose a different species.
Corey,
Yeah,
give it a go.
I agree with everything that Dave said. And I think for the projects that I highlighted here, we were really focused on developmental programming and the impact on adult health. And so if we were using macaques or baboons, you know, we'd be 20 years out before they reached, okay, maybe not 20 years, but 10 years out until they reach full adulthood and reproduction. Whereas with the marmosets, I have to wait 2 years and then they're reproductively viable. So, and then if you take that forward into aging projects, it's just a few more years. So the marmosets are really valuable for that, and they mature quickly, but they are closely related genetically. Their pregnancies are different than humans, but have similar characteristics, and the hormonal aspects are similar to humans. So I think they offer a real advantage, but I highlighted the baboon today as well, so that they had some time during the seminar series to show a different model, and they're obviously larger than the marmosets and take longer to mature. And so the developmental programming questions there have taken a lot longer to investigate, but they're a really stable model for this premature infancy. And taking these infants by C-section prematurely and assessing them, whereas no one that I know of has done that successfully with marmosets. So I think all of the things we've been saying in the symposium series that emphasize how important non-human primates are, as we face new critical health conditions, we need to understand and have the basic science for the animals and how they survive and live and what their characteristics are, so we can fit the best ones and not just go to a rodent or not go to C. elegans, that we understand the primates that are necessary.
So actually following up on that with the marmosets, you mentioned something that's always really puzzled me, and that is that they don't appear to have a lactational anovulation. Are New World primates the only non-human primates that don't have that? And if so, what is it about marmosets and other New World primates that allow them to act like rodents with a post delivery ovulation?
Yeah, so now you're asking me a taxonomy and life history question. I don't know who all can do this and can't, and I should know. I just taught primatology, but maybe Dave can pitch in here on this one. But to answer for a quick second, the marmosets have the adaptation of having a slowed embryonic development right after pregnancy, right after conception. And so the embryo is still growing, but it's slowed as the placenta grows. And there's a number of hypotheses for why this happens in marmosets. And part of this is that they're a prey species and they have to reproduce quickly, and they lose a lot of infants in captivity in the wild. And so evolutionarily, they have larger litters and produce quickly. But there's also hypotheses about how this then links to the chimerism question and placental development. There's a pause in development that allows the placenta to fuse. And the placenta grows first, and then the twins go through gastrulation and development. And the mechanisms underlying that and how that impacts development is still really unclear. And that's something, I mean, I brought it up at the end because I will always push marmosets as the best model for almost anything, but it's something we need to discuss as we develop them as models, whether they really fit and what the questions are that we're asking. And marmoset biology is just a little different. But Dave might have an answer to the others that are other species that ovulate after delivery.
With the South American species, not so knowledgeable on all the rest. The marmoset, though, it might come down to what you were saying, Corey, the energetic demand because of that slow gestation up until the suckling set of infants are really weaned may be key, because Alan McNeely showed in women that one of the key things that contributed to their post-birth suppression of ovulation was that energy demand from suckling infants. And as soon as that crossed a threshold of less demand, they started ovulating. And in the marmosets, that could well be the mechanism. And they've turned off because prolactin levels go sky high in marmosets, as you might expect, and they ovulate right through it. So they managed to turn that off, and it may be through that they have a way of monitoring, as we do, energy inhibition on reproduction. When there's such an energy demand in a woman's body, trying to get pregnant again is energetically a bad idea. So it's cut off. In the marmoset, it's not. So I think there could well be a lesson to learn there in the neuroendocrinology that would be very translatable, and the marmoset would provide a species that's had to conquer a niche and get over that we haven't had to do. So I think they could really open up an avenue there. That would be helpful.
Good. We're getting one or two questions. Unfortunately, Dr. Adams Waldorf is not available to answer those. I want to encourage anybody ASAP, if you have questions for Corey or Dave, go ahead and get them in, and I'll give it a minute. And while we're waiting for that, I can perhaps open it up to let Corey expound a little bit on a project that I know you are working on with Ricky Coleman, that is looking to do a retrospective study of the determinants of marmoset survivability in infancy. Perhaps you could tell us a little bit about what the problem is and how you're going about trying to research some answers to the problem.
In colony management of the marmosets, one of the problems is that infant loss is very high in the first 2 weeks. Marmoset infants have a much higher death rate than macaques or baboons. And this has been discussed in colony management for decades, and it's part of the grouping of behaviors that we kind of link as the marmosets are frail or difficult to manage. They all fall into that bucket. But people have argued for years that there should be a way to solve this problem and to figure out what characteristics can be assessed. Can we change husbandry? Can we change characteristics about the mother or the father? Or is it purely a result of these larger litters? So the marmosets can only support twins, typically, on their own without human support. We can do things like rotational rearing and cross fostering and nursery feeding to increase the triplet litters. But Ricky and I are looking at the health records from the 2 primate centers, Wisconsin and Southwest, that maintain large colonies. And we both find that survival to the first 14 days ranges in about the 60 to 70% numbers for both colonies, which means we're losing 30% of infants in a very short time frame. So whether it's related to the womb-to-womb type questions that I presented, or obesity questions, or whether it has to do with density in the room, maternal body weight, paternal body weight, parturition, how many pregnancies she's had. We're looking at all of those and factoring them together and actually looking for an epidemiologist to help with this. So if someone knows an epidemiologist that is really great with holy data and statistics, we'd love to talk to them about how to assess all of these variables, because we're looking from the maternal side and from the infant side to try to make recommendations for colony management and to be able to really expand marmoset colonies and success with those infants.
Okay, great. I think we may, because of our delays, and we probably exhausted everybody, and we're already past time to conclude. So I think we will. I want to thank our speakers so much for outstanding talks and for presenting the great work that they're doing and that their centers are doing. And I want to let you know that the next session is June 8th with Dr. Paul Johnson at the Emory NPRC is going to be moderating the session on genomics. So all of you be there or be square. And thank you all for attending and for your patience. And I hope you all enjoyed it. And we'll see you all the next time.